Kritika Gaukar. I'm the senior manager in Elucidator, and I work in the R&D team, and have been trained as a bioinformatics scientist, and I've worked at Children's Hospital of Philadelphia and Mayo Clinic previously, analyzing a lot of the patient omics and clinical data. And today I'm happy to introduce one of our features on our platform called PolyKG, which is our approach to co-build knowledge graphs to evolve with the specific use case and research needs for our customers. And this topic originated from our own experience of working with or utilizing traditional knowledge graphs, which are either off the shelf where the data is pretty generalized, so all the literature is curated to some level. The quality and accuracy could vary across the different sources that it's aggregated. The traditional way is also to manually process a lot of the data, which results in disconnected to emerging insights. So that means like maybe they've updated the database for Uniprot like 2 years ago. They've updated, a different CTD database like 3 years ago. So it's like varying in like the quality of the insight because it's not really updated frequently with the emerging insights and literature and research published in that domain. And also, in most cases, we see that molecular data is often neglected, mainly because it's challenging to model that internal molecular data that's very specific to your use case. So that's often like consumed outside the knowledge graph, which limits the return of investment of the knowledge graph, as well as like with all the inconsistencies across the processing and insight generation of that data, it really limits the novel insight that we really want to extract from the knowledge graph. And I'm curious, have any of you worked with knowledge graphs and faced the similar kind of issues? I'd like to know about those. And we can also like discuss about, how all of these have certain pros and cons. So for example, if you're manually processing a lot of the data, you have a lot of control on when to ingest that data and how to process and model that data. But really, without the infrastructure, it would take, too long to actually result or like appreciate the value of the knowledge graph. So I'll just wait for a few minutes or like a seconds, and then we'll see, what kind of results we come up with. Should we be, is this good, Nidhi?
Yeah.
Okay. Yeah, so you guys can like start polling, and we would then go into the what we've heard from other leaders in the domain, especially now that there's a lot of AI native approaches and strategies that a lot of the biopharma is thinking about, where they want their own data, their own indication, their own pipelines and science actually readily available in a knowledge graph or in basically a structured manner so that they can derive insights from it at scale. So some of the things we've heard from our partners and customers is about, can we build or integrate EHR data with the biomedical data from, say, TCGA and open targets to get more unique insights in the cohorts that they are interested in? We've also heard about how in manufacturing, a lot of the data that they produce is not readily available, or the literature doesn't exist in the unique cell lines that they use and the unique manufacturing processes that they use. So they would like to build a unique knowledge graph using that data, but also incorporate a lot of like the pathway databases and gene information to enrich their internal data, to rank targets that they might look into further. And we've also heard about how in biotech, there's a lot of like unique research and science people are studying that doesn't have a lot of published literature data. So aggregating a lot of literature data then doesn't make sense to this kind of organization. So they basically want to see how we can actually integrate their internal data that could be like thousands or millions of data points to create a custom knowledge graph that resides along with the limited literature that exists out there. So again and again, across different stakeholders and teams, we've heard that they need something that's customized, that is more tailored towards their use case instead of the off-the-shelf generalized knowledge graphs that are available out there, which are aggregating a lot of data, but not specific to their use case. So we believe that a collaborative way to build a knowledge graph is the way you can really evolve with your unique research needs. And that mainly means that you need to customize every step of the knowledge graph creation. So you would have to customize the data sets that's ingested at scale, the different modalities that you want to ingest. So it could be clinical trial data, EHR, or tabular omics data. And all of that needs to be harmonized uh, in a consistent manner, but that will also be custom for every use case. So that might be a different reference genome that you're using because you're studying like a really unique or novel protein. Or it could also be curation of very specific disease conditions and comparisons that you're interested in. So a lot of this machinery needs to exist, which is data-centric, to make the data more valuable or ready for knowledge graph. And then once it goes past this custom processing and very detailed annotation, the knowledge graph itself needs to be readily consumable. So traditionally, knowledge graphs was like a very useful tool, but you would have to query through different programming languages like Cypher, and that would be another like a learning curve. So that was something that hindered the use of knowledge graph traditionally. So now with AI and agents, you really need to make the consumption also customized to the different use case. So there might be biologists in your team who would want to ask questions in natural language. So that kind of infrastructure needs to exist so that now that the data is structured and available in a knowledge graph, biologists could ask simple questions about, is this gene associated with this disease? What are the drugs associated with? What are the pathways that this gene is in? And also extract useful inference from the internal data. So is it differentially expressed in the two cell lines that you're trying to study, or the conditions that you're trying to study, right? And the final level would be customization of the scoring. So this is an integral part that we feel is missing in most of the other knowledge graph solutions, where like every team has evolving use cases. There might be different indications that they might look at like 2 months from now. There might be different modalities that they are using to study a different drug or tissue. So this customization of ranking, which basically means that we have a way to integrate all the multimodal data to support, say, like a drug, a gene, or a pathway, so that you can have a customized way of scoring that replicates your thought process or your internal process. right? So instead of going through like thousands of genes, you would get like the top 5 genes that are really relevant to the way that your research is headed. And to support that kind of data infrastructure, right, like the data-centric infrastructure, you really need to be able to utilize AI agents or LLMs to scale the process. So Nidhi was mentioning about poly-extract, which is another agentic workflow where we are able to curate a lot of the data sets and literature at scale to get to level of depth that we are showcasing here. So we would start with the broad kind of data aggregation. So here's an example of like if you're interested in a target, you would want to get a lot of the gene-related data from NCBI, protein-related data from UniProt. Open Targets is a great resource that we would want to use to characterize targets. But you see, this is like the broader net of data. So any valuable knowledge graph would have this kind of broad, sources that it aggregates from. But we really want to capture the depth of the research that you're interested in. So we go with this tiered approach. For example, if you're interested in responder, non-responder characteristics of patients, or you would want to know what are the endpoints that people have used in similar drugs, what were the outcomes. So that would be like a level deeper, right? So it would be specific to your drug, indication, tissue, but that would be a level deeper. And then the most detailed information or valuable information would come from internal or proprietary sources. So this would be something that provides the deepest information that we can add to the knowledge graph. And that's usually from the internal data, the internal experiments that you run, internal data that is generated for the very specific gene knockout or drug that you're studying, right? So the knowledge graph that is built not only captures the broad areas of science, but also captures the depth from the very specific data that we either find from public domain or we aggregate from internal sources. And what that leads to is that once this data is aggregated and structured to get to this deeper insight that we were talking about, you can get really high quality triplets. So now this literature sources or the context that is needed from unstructured metadata around a data set is very available in a structured format. And you've introduced poly extracts, so I'll use that. So that's where you would get that kind of detailed structure from the literature. So that will help you identify triplets that can be extracted from the literature, but also like showcase during the curation process, where's the reference for this information? So is it the method section? Is there like a paragraph in the second page with this literature or this triplet mentioned? So a lot of the details will be captured or will be available for review for the user. So it's not like a black box that this triplet was identified, but you'll be able to trace back to where this actual information is captured in the paper. And like we were mentioning, traditional knowledge graphs have a limitation on how much data that they can add to the knowledge graph. They usually go with the raw forms, where it's just like the raw counts or TPMs, or the data is already processed like GWAS, where like we were mentioning, we know there's like a lot of value in the internal data and public data that's available there from molecular or omics data, but that needs to be processed. So for example, if you're trying to get more insights into specific genes that drive phenotypes in macrophages, this process would need actually pre-processing single cell data at scale. So a lot of data sets that have macrophage as cell types. We'd have to do cell clustering and labeling, as well as extracting informative genes. So that means not only differential expression, but also using a lot of the ML models to come to a list which is like the features which are important, and as well as differentially expressed in this phenotype that you're trying to study. Right, so I hope you appreciate like the agents, is like a full-on workflow where the data is actually processed to get to this triplet. And again, through the machinery, you could do this at scale. But throughout this journey where we were trying to understand like from our customers about their pain points, as well as where we need to um, head towards like the solution, we've learned a few lessons. The first lesson is mainly that ensuring high quality of that knowledge graph, while there are like different use cases that need to be tended to, is a very complex task that requires a data infrastructure that supports that kind of scale and quality assurance. So to showcase like what we've learned from our previous efforts is that we can't just like add a lot of literature and biological databases and create a knowledge graph. That's going to lead to very low ROI, mainly because the depth of the research or science that our partners are studying will not be captured in this knowledge graph. It will have incomplete metadata, so you'll not know like is this differentially expressed in macrophages or some other cell type without the context of like where this gene was studied. Um, it will have very poor quality linkages because the metadata doesn't exist to provide the context, as well as low quality. So overall, like even when the knowledge graph is created, a lot of data is ingested, the trust in the insights might be low because the user is not able to identify the context or trace it back to the source without the proper metadata around how that data was processed. So our approach, like I was mentioning before, is to create a whole data product. So that means that when we ingest the data as it is actually valuable. It's structured, findable, and quality controlled. So that means that this data product will have versioning. It will evolve as like the use case or like the number of data sets are available. So it's a product that product manager will manage, as well as like uh, you know like create a roadmap around like how will this data um, evolve with the science. Then we'd also have agentic workflow to identify how this knowledge graph or knowledge is extracted. Um, so that means like the entity recognition, like I was mentioning before, from the unstructured text that is done at scale. Relationship extraction is done in a way that it captures the context, as well as the processing details of that data type. And then finally, the knowledge fusion, which is the modeling of the data. Now that the data and the triplets are available, you really have to figure out how you model that knowledge graph. So how do you score those edges? How would you rank a specific node of interest given a disease of interest? Right, so these are like the three major things that we would want to use to create a knowledge graph that evolves with the data and science. And this knowledge graph then is customized to different use cases. So maybe you start with target ID, which is the most common use case that you've encountered. But then you want to progress into like what are the specific like subgroups that your cohort of patients are part of. Um, Maybe then there are like other teams who are interested in manufacturing or drug repurposing that also want to utilize this knowledge graph. So basically, this knowledge graph now needs to have all these custom relationships that will be used in a custom scoring framework. And all of this will have like a dimension of evaluation. So we follow these three dimensions. So for a knowledge graph that is created, trustworthiness would be, are the insights scientifically validated? For comprehensiveness, it would mean, is the biological context complete? And groundedness would mean that is it aligned to a specific biomedical ontology or standard? Some of like the evaluations that we've done for the knowledge graphs is around drug repurposing. So 4 out of 5 times we found that the drug disease connection for a drug that was repurposed recently in 2025 was available in the knowledge graph, which was closer than other, other unrelated diseases. So the valuable insights that are captured in this knowledge graph, on average, like 4 out of 5 times, will be valuable or significant insights. Most of the data that is aggregated from public domain is updated regularly. So as open targets or Campbell or Uniprot is updated, that's updated, the knowledge graph is updated as well. So we make sure that those sources are trustworthy, as well as the data is recent. And in comprehensive, we would say that each node has more than one relationship. So there is no node that is independent on its own or has very limited metadata associated with it. So each node that would be disease, drug, tissue, all of it would have more than one relationship to actually characterize that node. And for some entities of interest like disease, we really want to make sure that it's capturing the most deepest level of information, because a lot of it is available in like ontologies like Mondo. What is the subtype of this disease? What are the symptoms associated with it? What's the phenotype associated with it? So for each node, we really want to capture the most detailed version of relationships. So biologists could quickly query for those very nuanced connections. And like we were mentioning about groundedness being around biomedical standards, right? That's the whole process of creating a structured, findable, and quality controlled data product. So all the data that's ingested in the knowledge graph is mapped to ontologies that are publicly available. So, those are well maintained. There's a strict schema for each node and versioning, so that you can keep track of like what's changed from the different versions of the knowledge graph, what kind of data, what's the source of that relationship. A lot of the validation is part of the knowledge graph building process. The second lessons that we've learned is often, like we mentioned, that internal data is where the most detailed information exists about, the science that you're studying. But it's also where the data is mostly fragmented. It's available, but it's not useful for AI as it is. So that really hinders or like slows down the knowledge graph development. And that's because there are like different sources of that data. So some data could be generated in the lab. Some data could be generated from the CRO. There could be EHR data that you have aggregated from third parties, as well as process data that your internal computational biologists and data scientists have aggregated over time. So all of that results in data silos, large volumes of data being available, but really not being ready for consumption, like we've mentioned before. So our approach is to, again, go through the machinery to actually harmonize that data. So that would mean that unstructured text from like literature, internal study reports, tabular data, as well as EHR or EMR data goes through the harmonization process. So that means that it will go through the appropriate metadata harmonization. So you would want to map it to OMOP, for example, like EHR is mapped to OMOP. Then you have an ETL process to map it to the standard ontologies. Data processing would mean like we start from raw files for omics data and consistently reprocess it. So now that it goes through the harmonization engine, uh, it still needs to go through additional processes like bioinformatics or ML analysis to actually be ready for the knowledge graph. Because in the knowledge graph, we really want to capture the knowledge or the inference from the data. So raw data is great, but then it's on the user to like, again, reprocess the data to actually gain that insight. So for molecular data, we really make sure like the inference is captured. So for example, is this gene differentially expressed in the different cell types that you're interested in, or disease versus normal data sets that you're interested in? Are these features actually important? So like we run multiple ML models to figure out the top 100 or 600 features of interest, as well as do some network analysis to find out if there are genes that regulate different gene expression, right, to identify what are the driver genes in this condition. So then now all that inference is coded into a knowledge graph. So it's not just gene expression, but it's actually giving you information: is the gene differentially expressed in the conditions of interest? Is it driving that condition? Is it actually important for this condition that you're studying in? And that results in a knowledge graph that helps you get more insights than not just gene expression. But if you're interested in, say, co-expression of different genes, so it gives you the relationship, but also the probability that 40% of the time through our structural learning methods, um, these two genes will be co-expressed, or through it's differentially expressed with this p-value and fold change. So all that context from the molecular data is captured as properties um, for like a biologist or a computational biologist to query in the knowledge graph. And the third and the final lesson learned would be around how the data that is available across different sources, public or internal, really needs to be embedded into a knowledge graph through careful structuring and linking of those entities. So for example, like GTEx, right? It's a data source that is used very commonly to compare gene expression of like a disease condition to normal tissues. Um, However, if you look at the raw data, the data is quite large. It's like 27 gigs, and that would really result in low performance of the knowledge graph. And someone like a user that's using it would also again have to summarize between the sample level and tissue level to understand if the gene is actually expressed at the tissue level. So you really need to like carefully think of the use case and the consumption of that entity to figure out how that data looks like in a knowledge graph. For example, in this GTEx use case, we went from using from sample level to tissue level because that actually provides the information that's required for the use case. So that would be comparing the expression at disease level versus the tissue level, and also really reduces the amount of data that is actually ingested in the knowledge graph, so that now the knowledge graph is efficient and easily consumable. And that was just like one source. But in our knowledge graphs, on average, we are using more than 15 sources. So that would mean that you don't only model the data or relationships effectively, but you also have to come up with a weightage that helps the customer or the user rank the entities of interest. So for example, if you're interested in a gene based on the different drugs that it's associated with, you would want to identify what's the phase that the drug is in. So maybe if it's in the earliest stage of clinical trial, it's more useful or to work on it further because it's novel and maybe that's a better competitive edge that you can explore. Um, if there are a lot of drugs for that gene, maybe a lot of other companies are also like in that domain. But if there are fewer genes, then again, it's more promising and lesser competition, right? And then finally, like you would also want to know from literature as well as other databases that is this gene actually druggable? So are there approved drugs? What clinical phases they are in? Is this gene from like a druggable family? So all of this, like different sources of data, needs to be aggregated and structured in a scoring framework so that now biologists can ask questions about, is this gene actually useful for us or our pipelines? Yeah, and using all this approaches that we've talked about, as well as the data infrastructure, um, we've uh, we've actually delivered quite a few knowledge graphs to the customers. The one that we wanted to talk about in detail today was around this cross-species knowledge graph that we built for a non-model organism. So these organisms are very interesting to study in the context of human diseases, but using that non-model organism data from literature would result in very less data. So they wanted to utilize their own internal data that they have been aggregating over years. So now this knowledge graph needed to capture not only human-related information that is widely available in databases, but we needed to um, uh, aggregate as well as link the non-model organism data to the human diseases and tissues and genes of interest. Um, that currently was taking a lot of manual effort from this organization, and they really wanted to expedite this discovery process to reach a specific milestone in their company. Um, and that's where we partnered with them to create this unified knowledge graph uh, to not only harmonize and integrate their organisms' multi-omics data, but to really provide the context and the and democratize the data and the processed data insights through natural language based queries of the knowledge graph to their biologists. Um, and this is like an overview of what we were able to deliver to this organization. So it first started with poly KG V0, which is the base version. So previously, like we mentioned, the base layer of the pyramid is where you captured the breadth of the uh, domain that you're interested in. So it would be genes, drugs, um, diseases, um, like those kind of biological entities and their relationships are captured from public sources, and that's available on day one. So that means that a biologist is able to quickly query for, like, say, genes of interest and see if the gene, is currently in any clinical trials. Is it in a druggable family? So to create that hypothesis that they would want to explore later. Um, and the second step would be the pipeline development. So that means, like you were showcasing the pyramid, that means we are in the second and the topmost layer where we ingest public data as well as internal data. And this goes under not just ingestion, but harmonization, which is where we curate the data, we standardize the IDs, we also process this data. They had a pipeline that was currently very cumbersome or costly to run on that infrastructure. So we took on that pipeline to optimize it even further, to reduce the cost of the processing of that data. So we really were rebuilding and configuring our ETL pipelines to integrate the internal data and define the proprietary data model for these sources and data types. And that means that we've got AI-ready data product from the second step. And then the third step would then be the customization of the KG. So the organization had access to poly KG for over months. And then we were extending the knowledge graph schema to capture the custom relationships, the custom processed data or inferences from the data that they had shared with us and we processed. And basically going towards defining the relationships, the edges, and weightage of those edges across the different entities. So within 60 days, we were able to come up with a scoring framework for the specific use case and a customized knowledge graph. So like we were mentioning, that evolved over this process to use their specific scoring framework, to use their specific data and scoring pipeline was delivered, which they are currently utilizing. So the biologists as well as computational biologists are using the data through this customized knowledge graph now. And so this is where we are currently. So we have natural language querying, which helps with retrieving data. If a biologist asks a simple language query, the AI agents are able to create the cipher query, run the query, as well as return a summarized version of the results. But we are also expanding that capability, or like the AI capability, in our polyco scientists, where there will be a lot of reasoning and hypothesis generation that can also be enabled through the structured data that we've developed for them. And as an overview, we just wanted to showcase like the variety of data that we've used in this knowledge graph. So we were able to get access to a lot of variant data from their comparison of the non-model organism to humans, as well as comparative genomics and differential expressions of like different conditions that their non-model organisms were in. And that was enriched with human data, since eventually we want to learn about human diseases. So a lot of biological databases like Open, Targets, GTEx, UniProt, etc., were added as biological databases. Literature data related to the non-model organism, as well as other publicly available multiomics datasets, were all aggregated and harmonized through our harmonization engine. And that led us to create the customized knowledge graph and the target evaluation or scoring framework that they're currently using to do a lot of the target scoring and indication and pathway prioritization for their organism. All right. Yeah, and we have a quick demo about how this data looks like in the knowledge graph. So this is the NLQ consumption layer of the knowledge graph. So behind this, there are 51 million relationships, different nodes that are captured across disease, drugs, tissues, um, geoterms. So for example, if biologist is interested in learning about IL-13, and is it really associated with macrophage in any gene process or a cellular process? So biologists could simply ask, is IL-13 linked to geoterms, which contains macrophage? So it's pretty open-ended. You don't have to know the exact geoterm that you're studying, but you see the agent is then interpreting this English language query and creating a query which is also shown here, where it's capturing that the user wants to capture gene linked to a geo node where the gene is IL-13, and then to capture the sub graph related to this query. And it's also summarizing here that IL-13 is linked to gene ontology terms related to macrophages. Specifically, IL-13 is associated with positive regulation of macrophage activation. It provides like a description of that geoterm, which involves any process that stimulates or increases the rate of macrophage activation. So it's not just like a visualization tool, but it's really helping you summarize that information as well. So the agents that are working in the background are able to not only visualize this query, but also summarize the information from the metadata that's available in these nodes and edges. Similarly, you could also ask about the gene in general. So maybe there's a gene of interest that you're trying to study, and you just want to know what are the properties of that specific gene node. So in this case, the query would say the gene, where the name is IL-13, and result all the properties of that node. So here we are saying the gene name, gene synonym, gene antibody tractability, symbols, locations, etc. A lot of the different properties that we've captured for one node. So this this is where we want to showcase that even one node has a lot of different properties that are captured in this knowledge graph. So for example, this is IL-13, but it's captured, there's different properties across the different organisms. So we really will have to scroll through this to get to our the human organism. So I'll quickly try to get to the human row. You could have also just asked for IL-13 properties in humans, but I just wanted to showcase that there are different organism-related information. So for example, here, this row is IL-13 and 9606, which is our human taxonomy ID. It's providing insights into, is the gene druggable as an antibody? So it says it has approved drugs. The geo term associated with the cellular component is providing it high confidence because it's at the cell surface or membrane. It's also capturing other drug abilities. So is it protein tractable? So are there like protags associated with this gene? And it's also capturing the location of the genome as well as some IDs that we've curated. And that mainly will give you an overview of one's node in more detail. And this again goes towards what we were saying, that we don't want to capture like not only the breadth of the domain and science, but also the depth of each node and relationships. So now that we know that this gene is druggable, you could just again ask in plain English language, what are the drugs associated currently available for IL-13. And now it would query a different relationship. So it's querying gene that targets, that is a target for a drug, and then returning the result in this kind of a summary. So it's captured a lot of different antibodies that the drugs that are associated with IL-13. They've mentioned some of it here. These are the medication part of ongoing efforts to modulate the activity of IL-13. Again, like summarizing some of the properties of IL-13, where it says a gene that's impacted various different inflammatory or allergic response kind of conditions, mainly because of the different metadata associated with this IL-13 gene. So we've captured how we can look at details of an IL-13 or like a node, but also various different kinds of relationships from different sources. But like we were mentioning, the most important or detailed relationships would come from the molecular data that's actually relevant to the use case or internally produced, right? So we can't showcase the internal data, but as a proxy, we've showcased that if the use case was to learn about healthy versus diseased conditions, and is IL-13 really differentially expressed in these conditions, the knowledge graph would be able to query this information through, like if you ask for natural language query around is IL-13 differentially expressed in any comparison containing healthy and disease, and then list all the p-values and fold change, a comparison ID may need to provide the context and details of this inference. The query that's generated will use the gene ID and then the differential expression relationship with the tissue, and identify that the comparison contains healthy as a term. So this can only be possible when you've actually curated this comparison, right? Like you might appreciate that only when this term is harmonized across different sources, different data sets, you'll be able to query this effectively. So this showcases that the data that is ingested in the knowledge graph needs to actually be very highly and deeply curated so that it can be consumable. So it's like querying for this term healthy across the property of that relationship and identifying the actual molecular insight or inference that IL-13 with this p-value is differentially expressed in the comparison of interest. And again, the summarization agent is kind of capturing the details here that IL-13 is differentially expressed in several comparisons. Notably, it's capturing that there are different data sets that have studied asthma and healthy conditions, and it's summarizing this table basically. So if someone's new to differential expression or any of like the processes that you've processed the data through, it will summarize some of the inferences as well, mainly to enable that consumption of the data. Right. Okay. Oh, okay. Yeah, and then to summarize the whole conversation that we've had today, it's mainly to say that we believe that there's actually a need to co-build these knowledge graphs for biotech. And a lot of the knowledge graph solutions or approaches are limited in the data sources that they use, or limited in the capability of integrating internal data. Also, without like the appropriate data infrastructure, a lot of the traceability, the trust in the data is reduced, um, which then like reduces the consumption of that knowledge graph. And finally, we want to also emphasize that like each user, has their own consumption methods and layers. So if the knowledge graph needs to be valuable and something that really is used across the organization, AI will be needed to consume the amount of data that's aggregated in the knowledge graph. So the infrastructure not only has to capture or enable creating the knowledge graph, but also consumption of that knowledge graph effectively. And all that means that there's a lot of customization that needs to be part of creating this knowledge graph. And so we talked a lot about how we're creating customize or co-building these knowledge graph. But what we envision is that this will eventually be a co-scientist that is developed specifically for this organization. This is in our 6-month roadmap, where we're using LLM for data, which is basically tools like polyextract that Nidhi was mentioning, or that help us, curate deeply. That becomes like the first layer of how well that data is curated, how deeply it's curated to make it valuable, which then goes into data for LLM. So that means now this data that is curated, standardized, and available actually is provided to LLMs to structure in a way that is useful for visualizations, or agents that do data retrieval, or agents that query knowledge graphs like you were showcasing. We've already built a lot of these agents. So within the 6 months period, we'll be launching our co-scientists, which enables all of this agentic workflows through simple non-code interfaces like we've shown in the previous slides, to enable the expedited or faster consumption of data and faster insight generation eventually. Right. I think that's all from me. Thank you so much for listening to me over the whole time that I was talking. Hopefully you've learned a few things, and yeah, we'd love to hear back from you. You can email me or Nidhi, and we'd like to learn more about your approach or your research needs. Yeah, thank you.
Thanks, Kritika. Before we leave, there are a couple of questions. Right.
Yeah.
So, one of the questions is, can we define the weights of the edges?
Yeah. So in this, oh, 1 second. So in this example, the weights of the edges were defined according to this framework that we've used. So this target gene had all its different uh, factors that were used. And each source would have a specific weightage. So this relationship would have an eventual score, which would be from 0 to 1, which is calculated through this different sources as well as scores associated to the actual values of the relationship. So then what happens is that you can rank the gene with the score associated with it. So if it's highly ranked across these different sources, the score eventually for this relationship would be higher than something that's less important in this scoring framework. that is the final goal. Yeah.
Okay. Another question, is PolyKG custom to each organization, or can it be deployed internally?
Yeah, so we provide both those offerings or solutions. A lot of the POCs or the, to showcase the capability of the knowledge graph can happen in our Poly platform, which is SOC 2 HIPAA compliant. But we are also able to deploy the platform in the organization's infrastructure. We've got a lot of engineering support and product support to enable that as well. Yeah.
Okay. Last question. What are the query limits, and do the facilitate example turning large chunks or the whole knowledge graph into a matrix in a notebook for algorithmic processing?
Yeah, so that's something we are working with EHR data, since that's a lot of data that needs to be aggregated. So the whole knowledge graph in that context is created as embeddings with specific matrix or like vector values that can be used for, like patient clustering or EHR clustering kind of exercise or use cases.
Okay. So no more questions in the Q&A box. Thank you so much, Kritika, once again, in enlightening the audience about this, about our PolyKG. And thank you so much, everyone, for taking the time out to attend this webinar. We look forward to having you in yet another webinar on August 28th on Poly Extract. Yes, thank you so much.
Great.